The EpiNext ISAC preparation kit is a complete set of optimized reagents designed for preparing in situ accessible chromatin and identifying open and closed chromatin regions. The kit offers a fast and versatile approach to chromatin accessibility analysis. The entire procedure can be completed in just 30 minutes using an in situ workflow that helps preserve chromatin structure. Sequence-independent cleavage helps minimize transposase-related fragmentation and GCAT bias, and the prepared chromatin is compatible with both gene-specific and genome-wide analysis. In the assay, cells are collected and permeabilized. A specialized nucleic acid cleavage enzyme mix fragments the chromatin and removes exposed DNA at accessible regions, while protein-protected DNA remains intact. The protected DNA is then purified for gene-specific analysis by real-time PCR or genome-wide analysis by NGS. The kit includes all components needed for in situ accessible chromatin preparation. First, prepare working permeabilization buffer by diluting protease inhibitor cocktail with permeabilization buffer. After collecting and washing your cells, resuspend the cell pellets in the working permeabilization buffer. To the resuspended cells, add nuclear digestion enhancer and cleavage enzyme mix. Rotate at room temperature for 6 minutes, then stop the cleavage reaction by adding cleavage stop solution. Spin down to pellet the cell debris and transfer the supernatants to new PCR tubes. Add proteinase K and incubate at 55 degrees Celsius for 12 minutes in a thermocycler. Add DNA purification solution, followed by 100% isopropanol. Resuspend the DNA binding beads by vortexing. Add the resuspended beads and mix thoroughly by pipetting up and down several times. Incubate at room temperature for 4 minutes to allow the DNA to bind to the beads, then place the tubes on a magnetic stand until the solutions are clear. Carefully remove and discard the supernatants. Add freshly prepared 90% ethanol, then carefully remove and discard the ethanol. Repeat once for a total of 2 ethanol washes. Resuspend the beads in elution buffer and incubate at room temperature for 4 minutes to release the DNA from the beads. Capture the beads by placing the tubes on the magnetic stand until the solutions are completely clear. Finally, transfer the solutions into new PCR tubes. The purified DNA can now be stored at -20 degrees Celsius or directly used to construct libraries for sequencing by proceeding with the end polishing, adapter ligation, and library amplification sections of the ISAC-seq protocol. With the EpiNext ISAC preparation kit, you'll achieve fast, sensitive, and specific mapping of chromatin accessibility. For more information, visit epigentech.com or contact your local distributor.